Alright, welcome to video 2. We are continuing our tour of the skin. We are currently in the dermis, but now we are talking about derivatives of the epidermis. These are tissues that started out on top of the skin, and then as it developed, they invaginated or grew down into the skin and became things like glands, hair follicles, and nails. Glands are really what we're going to spend most of our time on. So first up are the sweat glands. We're going to have two different types of sweat glands. The kind that you saw in lab is the eccrine sweat gland. These are for thermoregulation. They secrete mostly water along with some salt and nitrogenous waste. The salt and the waste are there so that you can get the water to move. Right? You pump the salt out of the cell, and then you leave all the water behind and you create a hypotonic environment inside of the cell, and then water osmoses out of the cell into the gland duct itself where all of the solutes are. So when you're making sweat, what you're doing is pumping waste and salt and then getting the water to follow by creating an osmotic gradient. So that's why the waste is there, not because you necessarily need to sweat to get rid of it, but you need to get rid of it in order to sweat. You have to pump something to get the water to move. This is what the sweat glands look like. They are simple cuboidal and sometimes stratified cuboidal epithelium, so they're square or roundish cells stacked on top of each other. Then you have a different type of sweat gland called an apicrine sweat gland. As this says right here, they are combined to the axillary and anogenital region, so armpits and around the genitals. They basically produce sweat plus some other stuff, is what you need to know. And this is where, this is where your body odor comes from. So sweat itself is odorless, as is most of the stuff that you find in it. But the fat and the proteins that you find in this apicrine sweat are tasty or raw materials to bacteria. So it's the bacteria that eat the substances in your apicrine sweat that give you your particular body odor. In addition to apicrine sweat glands, you also have these two other glands that are derived from apocrine sweat glands. So the wax glands in your ear, or ceruminous glands, as well as the mammary glands that you find in breast tissue. Then you have the sebaceous glands, which you've seen before. So this is a piece of information you want to keep in your head for lecture as well as lab. Eccrine sweat glands for thermoregulation, sebaceous glands for oil secretion. And this oil is antimicrobial or bactericidal, so it inhibits bacterial growth, but it doesn't completely prevent it. And then it's also there to soften your hair and skin. And remember, this is so that your skin doesn't crack and become an avenue of infection, not because softer skin looks better. Then you have hair. We're not going to worry too much about hair. Really, you just want to know that it's dead keratinocytes. So the cells that make up your skin are the same thing that you find in hair, only they're compacted much more and they have melanin within them. And it's different ratios of one type of melanin versus another that leads to blonde hair versus brown, red, or black hair. And we'll leave it at that. Different types of hair I could honestly care less about, and you probably don't care either, so we're going to skip this. This is why it's like gray. We also don't care about alopecia, which is spot baldness versus true or frank baldness or male pattern baldness, whatever it's baldness. We do care about skin cancer, though, so this will slow down just a little bit and talk about. So the risk factors, as this says here, include UV radiation and frequent irritation of skin. Whoops, I skipped ahead and I didn't mean to. We are going to go over 3 different types of skin cancer. For each skin cancer, you want to know which cell type or which cell layer it originates in, so which cells actually become cancerous, and then be able to rank them in terms of their aggressiveness, or how, not necessarily how quickly they grow, but how good they are at spreading from the epidermis down into the dermis. So it's not really the size of a tumor that's a problem when it comes to cancer, but how well those cancerous cells can move from one tissue into another. And you're going to see when we do the case study, this involves disassembling that basement membrane so that you can make it from the epidermis down into the dermis. That's why we spent time talking about it. At any rate, first up, basal cell carcinoma. This originates in the germinating or mitotically active cells of the stratum basale. It is not surprising that this is common because you're more likely to get mutations and problems with regulating mitosis in a mitotically active cell population like the stratum basale. Fortunately, this is also the least aggressive, so it doesn't usually lead to full-blown cancer that would invade other tissues and affect your health. This right here is an example of a basal cell carcinoma. It tends to be more prominent on the nose and face. I'm not sure why. Probably UV exposure on those parts of your body. Then you have squamous cell carcinoma. This arises in the keratinocytes of your stratum spinosum. So generally these are cells that don't divide, and because of mutations that they've developed, they start dividing when they are not supposed to. This is, as it says down here, more aggressive than basal cell carcinoma, but still not very good at spreading out of the epidermis and into the dermis or other tissues. So generally you remove it and you can get rid of all the cancerous cells and you don't have to worry about it. So this is what squamous cell carcinoma looks like on somebody's scalp. It may not be dangerous, but it sure is ugly. Then we go to melanoma. This is the one we need to worry about. This arises in the melanocytes of your stratum basale. It is, as this says right here, highly metastatic, which means these cells will leave the epidermis, crawl, like physically move themselves into the dermis, make their way into a blood vessel, and then pop out of the blood vessel somewhere else. So it's got quite a few skills that these cells have to develop in order for them to leave the tumor, get into the bloodstream, and set up shop somewhere else. So this we spend a little bit more time talking about. You want to be familiar with the ABCD rule. So this is how you evaluate a mole to tell how likely it is to be melanoma versus just a simple mole. So A stands for asymmetry, and it can be asymmetry either in shape or size. So if it's not a circle, or if it's got, if it's not a circle, or it has a very irregular border, or the pigment isn't even, then that's not a good sign. If you have an irregular border, so circular, oval, or otherwise, if the border has a lot of indentations and invaginations, this is more indicative of melanoma. Generally speaking, the darker it is, the worse it is. And then also, again, if you've got dark and light and sort of a splotchy or patchy appearance to it, that is very bad, or that is a bad sign. And then if the diameter is bigger than the size of a pencil, that is likely that it is no longer a mole, but it is now becoming a melanoma. Heads up, gross picture next. This is a melanoma on somebody's toe. So if you look at it, the color is not symmetrical. It's no longer a circle. It's got a really uneven border. It's too big. It's got dark spots and red spots, and it's scaly. All of these are bad things. If it's small, round, reddish brown, and not too dark with a regular border, then it's just a mole, but this is ugly. Then we get into burns, the last thing we have to do. So basically you just want to know the difference between first degree, second degree, and third degree burns. We're not learning how to treat them. This isn't really a clinical course, but we should go over the basics at least. So first up, first degree burn means damage to the epidermis only. So you've got redness, maybe a little bit of swelling, and yes, of course, some pain. Then a second degree burn means it's the epidermis and possibly some of the dermis, and this is where you get blistering. So the blister is when your epidermis is separating from your dermis. So you're getting damage to the basement membrane, I believe, and separation of the tissue layers. So here you have some ugly looking blisters down here and some redness and first degree burn up there. Then you have your third degree burns or full thickness burns. So this means the epidermis is gone, most of the dermis is gone, and you've got damage down into the hypodermis or below. With a third degree burn, you can actually get some reddening or some blackening of the skin. Oddly enough, these burns tend not to be painful at first because you've destroyed all the nerve endings. But when they do heal, they are very painful to heal. And one of the... I can't speak. I've lost my ability to speak. One of the things you need to worry about with a third degree burn is dehydration, because you've lost your epidermal barrier to evaporation. So that stratum granulosum is gone, and you can lose a lot of water in a short period of time after a third degree burn. And for these, they have to replace the skin with something because you've lost all of your epidermis and most of your dermis, and that doesn't grow back. I won't show you the picture of the third degree burn because it's gross, but that is the skin. We're done in 2 less than 15-minute videos. Thank you for listening. So then when we pick up on Tuesday of next week and I see you all again, we will be on to bone. So this is it for skin, and we'll talk about homework and case studies and everything when I'm back on Tuesday.